Hello everyone. My name is Shloka Krishna and I am a senior at the Foundation School in Karnataka, India. Today, I'm excited to present my research on how herbal extracts can act as potential senolytic agents in chemotherapy to target inflammation and senescence, hence improving patient outcomes. The reason I chose this research topic is because I was inspired by my grandmother's breast cancer journey to find more natural, holistic treatments for cancer therapy. Also, Indian Ayurveda, an ancient health system that relies on herbal formulations, has a great but overlooked potential in treating cancer complications. Now, before I dive in, here's a brief overview of what I'll be covering. I'll start by introducing cellular senescence and how it relates to aging and cancer therapy. Then I'll share the objective of my research, explain the methodology, and discuss the results I found. We all know how aging works. We live through it every day. But have you ever wondered what is the deeper cause of aging? That's where senescence comes into the picture. Senescence is the permanent withdrawal of cells from the cell cycle. Cells remain active, but they stop dividing. What's interesting is that senescence and aging are interconnected. As we age, senescent cells accumulate, contributing to tissue dysfunction. However, senescence isn't just a byproduct of aging, it can actually drive aging itself. When stem cells undergo senescence, tissue regeneration declines and this speeds up the aging process. In fact, studies in mice show that carefully controlling or removing senescent cells can promote what's known as healthy aging. And that's where cancer therapy comes in. Chemotherapy often induces senescence in cancer cells, which can be both helpful and harmful depending on how we manage these cells. Now that we understand the basic definition of senescence, let's look deeper. Senescence begins when a normal cell experiences stress, like telomere dysfunction, oxidative stress, or mitochondrial damage. In response, they stop dividing and enter a protective state. However, these senescent cells secrete inflammatory molecules called senescence-associated secretory phenotypes, or SASPs. While SASPs reinforce senescence in nearby cells, they also create a pro-inflammatory environment that can be harmful in the long run. This becomes extremely important in cancer treatment. Chemotherapy and radiotherapy aim to reduce and destroy cancer cells, but some of these cells enter senescence due to therapy-induced stress. At first, this seems helpful, but if these senescent cells aren't cleared, the SASPs they release can promote inflammation, weaken tissue, and even encourage cancer reoccurrence. As I explained earlier, senescent cells secrete SASPs. SASPs are made up of cytokines, growth factors, and proteases that influence its surroundings. They are responsible for either promoting or preventing cancer progression. On one hand, senescent cells play a protective role for normal cells. They recruit immune cells and secrete molecules such as PAI1, IL-6, and IL-8 to induce cell cycle arrest and therefore prevent cancer cell progression. On the other hand, in abnormal cells, such as cancer cells, senescent cells could lead to cancer cell progression. They could secrete pro-inflammatory cytokines like IL-6, IL-8, and IL-1 alpha that will cause inflammation and therefore lead to cancer cell progression. Other factors can also contribute to this process, such as MMP, EMT, and growth factors, but I will focus on the main cytokines I have analyzed, IL-6 and IL-8. From the diagram, we can see that IL-6 and IL-8 can act as both a pro and anti-inflammatory molecule, depending on its levels and context. My herbal formulation HF1 has 5 key components: cinnamon, green tea, turmeric, cardamom, and clove. Each contains senolytic agents that target and kill senescent cells. Green tea, for example, contains two important compounds, epicallocatechin gallate or EGCG and quercetin. Both are known to reduce inflammation and eliminate senescent cells. Turmeric, on the other hand, has curcumin, which is known for its antioxidant, anti-inflammatory, and anti-diabetic effects. As we age, defective mitochondria produce excess reactive oxygen species, ROS, which triggers oxidative stress and accelerates senescence. This leads to inflammation, metabolic issues, and overall unhealthy aging. However, interventions like caloric restriction and senolytic agents such as quercetin and curcumin can clear these senescent cells and restore mitochondrial health. By reducing senescence, these agents promote healthier aging and potentially even an extended lifespan. Now let me walk you through this flowchart to better understand the sequence of events in cancer treatment and how senescence plays a dual role. When cancer cells undergo standard therapy like chemotherapy or radiotherapy, they experience significant DNA damage. This triggers cellular senescence, either with or without the activation of important genes like p53, p21, and p16. Now, there are two potential outcomes at this point. First, senescent cells can have a beneficial role. They may trigger an immune response that leads to their clearance and ultimately cell death. This helps in reducing the chances of cancer relapse. However, there is also a negative outcome. Senescent cells secrete SASPs that can create a tumorigenic microenvironment. This environment can support cancer cell survival and even promote cancer stemness, where cells regain their ability to proliferate. As a result, this can lead to tumor relapse and further disease progression. My objective was to evaluate whether my herbal formulation HF1 can mitigate chemotherapy-induced inflammation and clear senescent cells in breast cancer cells MDA-MB. Specifically, I wanted to see if HF1 can reduce the levels of pro-inflammatory cytokines and expression of inflammatory genes in these cells. Next up comes the steps I took to fulfill this objective. The first step was to perform a cytotoxicity assay. Cytotoxicity refers to the ability of a drug at different concentrations to cause damage to cells, leading to their dysfunction or death. Cytotoxicity assays help assess the effects of the drug on cell viability. I used this technique to find the IC50 values, or the concentrations at which 50% of the cells were killed. After trypsinizing and counting the cells, I cultured MDA-MB breast cancer cells and treated them with varying concentrations of HF1, as well as with the standard chemotherapy drug 5-FU, both individually and in combination. After 48 hours of treatment, I added MTT dye to each well. MTT is a yellow dye that turns purple in the presence of live, metabolically active cells because it binds to mitochondrial enzymes to form formazine crystals. The intensity of purple color is directly proportional to the number of viable cells. By measuring the absorbance of this, I was able to calculate the IC50 values. This helped me understand how effective HF1 and 5-FU are in reducing cancer cell viability. The remaining cells were cultured in a 6-well plate and further used for gene expression studies. Next, I performed cytokine analysis. For this, I used 6-well plates. I plated 0.2 million MDA-MB cells per well across 5 different treatment groups. The control, MDA-MB and 5-FU, MDA-MB 5-FU and HF1, MDA-MB and HF1, and MDA-MB and DMSO. The cells were treated with their respective drugs, and after the incubation period, I collected the supernatants for cytokine analysis. Using a standard ELISA kit, I measured the levels of IL-6 and IL-8. This helped me understand how HF1 affects IL-6 and IL-8 levels. For the final part of the methodology, I investigated the expression of specific genes involved in inflammation. With the remaining cells from the cytokine analysis, I used the trisol method to isolate RNA from the cells. The isolated RNA was then converted into cDNA through reverse transcription. The cDNA was used for PCR to measure the expression of two genes, GAPDH, a housekeeping gene that was used for normalization, and COX2, an inflammatory gene that is upregulated in response to SASPs and is linked to cancer cell progression. Looking at COX2 levels helped me gauge HF1's potential in reducing inflammation. Now come the results. As I described before, we used the cytotoxicity assay to find the IC50 values of our drugs. Here, image A shows the trend in 5-FU treated cells with the IC50 value of approximately 17. As the concentrations of the drug increased, the cell death observed also increases gradually. Image B details the trend in HF1 with an IC50 value of 6.75. A similar trend is observed in this case as well. I also looked at cell morphology. In control cells, these cells looked healthy and well spread. With 5-FU treatment, the cells became granular and enlarged. HF1 treatment alone led to smaller, round cells, fewer in number, which was evidence of cell death. The combination of 5-FU and HF1 led to an even more cell shrinkage and irregular shapes, showing strong cytotoxic effects. Moving on to my cytokine analysis result. The graph shows the x-axis, which contains all the groups I used, and the y-axis, which shows the fold difference across these groups. In this graph, the MD-AMB treated cells with 5-FU show a significant upregulation of both IL-6 and IL-8. HF1 is effective in bringing down both cytokine levels on its own and in combination with 5-FU. Finally, I analyzed the expression of COX-2. Figure A shows that COX-2 was upregulated when cells were treated with 5-FU, but when treated with HF1, either alone or in combination with 5-FU, COX-2 expression was significantly reduced, hence reducing inflammation. In image B, we can see the PCR gel image for GAP-DH. The similar intensities of the band indicate that our samples were reliable and consistent. Image C shows the PCR gel image for COX-2. The brighter bands in the 5-FU treated lanes indicate higher COX-2 expression. However, when cells were treated with HF1, either alone or in combination with 5-FU, the intensity of these COX-2 bands decreased significantly. This supports the data in image A, confirming that HF1 effectively reduces COX-2 gene expression, hence reducing inflammation. To sum up, HF1 showed good cytotoxic effects on breast cancer cells. It reduced the secretion of pro-inflammatory cytokines IL-6 and IL-8, and it downregulated the expression of COX-2, a key inflammatory gene. These results show that HF1 can potentially be used as an adjuvant therapy to help prevent cancer relapse. For future studies, I plan to perform beta-galactosidase assays to assess cellular senescence further. Here are my references. Thank you so much for listening.